All right, so today we're looking at the experiment gamma tubulin is present in Drosophila melanogaster and Homo sapiens and is associated with the centrosome. This was done by Zhang, Zheng, and Oakley. So the objective of their experiment was to determine if gamma tubulin is a universal protein, meaning that it's found in many different species, or if it only is found in specific species. So some background information on this is that, well, first of all, Y will represent gamma and B will represent beta throughout this presentation. So gamma tubulin was first found in Aspergillus nidulans, and the gene that it codes for was called MIP-A. And so obviously by its name, gamma tubulin is part of the tubulin superfamily, so it's just like alpha and beta tubulin. Gamma tubulin is also associated with the centrosome, the spindle pole body, SPB, which was discovered after they found gamma tubulin antibodies on the SPB, and the SPB is something that is found in A nidulans, so that's what the SPB is. And after they first discovered gamma tubulin, they thought that it might have possibly been the long-lost -N nucleator mitosis, which they wanted to find for so long. But even after they found it, the first question they had to ask, though, was, is it found in just A nidulans, in close related species to it, or is it found in many different species, say like humans and things like that? So what did they do? Well, the first thing that they did is, well, I like some background information first, is that they used two cell lines. They used HeLa cells of Homo sapiens and Drosophila melanogaster. So obviously it's HeLa cells from humans and Drosophila melanogaster is fruit flies. And what a HeLa cell is just an immortal cell. So it's just an immortal human cell and an immortal fruit fly cell. They also did look at a mouse cell line that was called 3T6, but that's not as important as the human and the fruit fly ones are. So what first is they used cDNA. Now, and cDNA is just complementary DNA, which is just RNA that has been reverse transcribed into DNA. So anyway, so they used cDNA from A nidulans to find a sequence from fruit flies that had a 67.4 common sequence for amino acids, which was very important. And then they took that same cDNA sequence from the fruit flies and used it to find a cDNA sequence in humans at a 78.3% common amino acid sequence, which from those two things alone, that told them that this has to be the exact same gamma tubulin gene. And what this did is that it told them that the gamma tubulin gene is actually present in more than just the fungi, which is what A nidulans is, it's a fungi, which wasn't very important. But now they needed to know also after this is, well, is it actually expressed? So what they had done is they had stained the cell lines with gamma and beta tubulin so they could actually be visible during mitosis. And I'm not going to go into detail here, but one thing to mention is that obviously because they had just discovered gamma tubulin is that they went through a purification process after finding it before they actually stained it, because they needed to make sure that they actually had gamma tubulin antibodies would only bind to gamma tubulin. Because throughout what they were doing is that they found that gamma tubulin might have been associating with the beta tubulin binding sites. So they needed to make sure that they went through this whole long purification process, which is why they stained it with gamma and beta tubulin. So it's very detailed and complicated, so I'm not going to go through it here, but I will mention that they did go through a purification process. So the results of after staining everything is that they were able to observe gamma tubulin in the cells during the mitosis. And one thing they had noticed that the amount of gamma tubulin was directly correlated to how much microtubules and centrosomes were involved during that specific phase of mitosis, which was very, very important. And one thing that they had noticed is after looking at gamma tubulin in all the different phases, they noticed that the staining was the most intense and most visible in prophase. So prophase, prometaphase, and metaphase. Those were the most intense phases that they could see it. And obviously, but the key takeaway from all of this, though, which helps to prove a gamma tubulin is different from beta tubulin, which was a problem they had in the purification process I mentioned earlier, is that gamma tubulin, when I show you the gels, not gels, but what they used, is that gamma tubulin stains different parts and you see different things than you do with beta tubulin, which is very important. So here are the images that they had produced. So the one on the left, so first of all, A and D in both images are just phase contrast micrograms, so they're not that important that we're looking at here. But first we'll take the one on the left. So on the left, B shows the chromosomes stained with DAPI, and C shows the same cell or same cells stained with gamma tubulin. And keep in mind that B and C, throughout all of the images I'm going to show, B and C are always going to be in metaphase. So C is in metaphase. All this was doing was showing that gamma tubulin is associated with the centrosome, which is where you would expect it to be during metaphase, obviously, when the chromosomes are all lined up as they are. And what E and F on the left represent is that, so the images on the left represent cells in telophase, and the ones on the right represent cells in prophase in E. And again, all E is showing is just the chromosome stained with DAPI. And then F, which is very important here, though, is that F is again stained with gamma tubulin. And hopefully you can see the arrows on the bottom on the left, hopefully in F, but it's pointing to where the gamma tubulin is. But the thing is that in telophase, obviously, the microtubules have pretty much have done everything they had to do. So their activity and centrosome activity is not very high by that point. So the amount of gamma tubulin you would expect to see is obviously very low. And the one on the right does show the prophase, which is a lot brighter, which you'd expect it to be, because in prophase, that's when a lot of the microtubule arrangements and things like that are happening. So let's go to the one on the right. So the one on the right is probably the most important one, because the one on the right is showing cells stained with beta tubulin and gamma tubulin. So again, like I said, A and D can be ignored. B and C is in metaphase. So B represents beta tubulin, or cells stained with beta tubulin, and C represents the cells stained with gamma tubulin. And again, which is important here because you're seeing two completely different things. And B, you're seeing more so like the chromosomes and things that are being moved or beta tubulin is binding, whereas C, you're seeing specifically the gamma tubulin being associated with the centrosome, which is what it was supposed to do. And you can see that obviously they're two completely different things, which is the most important part of gamma tubulin, or this experiment, is to prove that they're two separate proteins. And then what E and F are showing is metaphase and anaphase. And again, E is showing the cells stained with the beta tubulin, and F is showing gamma tubulin. And again, even though you don't see anything in F, the most important takeaway from this is that, again, you're seeing two different things, which provides you more evidence that these are two different proteins that have performed their own roles during mitosis. And then the mouse cell line, which isn't as significant, but I'll still show it here, is that, again, so first of all, B and C, again, A and D are nothing more than phase contrast micrograms, so those can be ignored. So B and C, so B shows, again, the beta tubulin, and C shows the gamma tubulin. And all this is trying to show is that in animals in general, that the gamma tubulin likes to associate itself around the centrosome mainly. So that was the most important thing with the mouse cells. So again, B is beta tubulin, C is gamma tubulin. And if you look at the bottom, E and F, so E and F actually represent cells in interphase. So E, again, is stained with beta tubulin and represents interphase. F is stained with gamma tubulin and obviously represents interphase. And you can see, obviously, on the left, you can see two dots, which they had assumed is representing that the centrosome has duplicated itself. But again, the main takeaway from this, again, though, is that gamma tubulin and beta tubulin are showing you two different things when you stay in the cells in the same phase, which, again, is ultimate more proof and evidence that gamma and beta tubulin are their own unique proteins. So the conclusion and takeaway from all of this is that gamma tubulin is its own unique protein. It has its own role in mitosis, and it's separate from beta tubulin. But again, it is also, again, part of the tubulin family, but again, it does its own unique role. But then another thing that they looked at, though, is considering the three species that they examined, right? Fruit flies, humans, and fungi. They went to the conclusion that, well, gamma tubulin is probably a universal protein, at least in terms it's found in all eukaryotes, which means that it will play the exact same role in mitotic cell division, which is very important. And this was supported even further after they had duplicated Xenopus gamma tubulin genes. So they didn't explain anything further with that, but again, they did mention it, which also further supports it. And the last thing, another takeaway from this is that gamma tubulin is found at the centrosome of animals. It's found at the SPB of fungi, which also suggests that it's probably a major component of the microtubule organization complex, which is very important because again demonstrating that gamma tubulin is associated to a different place and doing different things than beta tubulin, which is ultimately, and also again that it is found in more than just a fungi and related fungi species is found in all eukaryotes, which also was the main takeaway of this experiment.